are very proud إنه ولادنا from everywhere in Egypt are يعني contributing today, and I thank them very much for taking this step. حتى لو هم بيحاولوا for the first time, سواء نجحوا أو ما نجحوش, they have to keep trying, this is an excellent exposure for them.
انا الحقيقه ابريشيت كلهم بصراحه يا دكتوره الهام، فعلا يعني الحقيقه يعني انا مبسوط ان يبقى في ناس تراينج، يعني لان ده بيدي فرصه ان هم يحسنوا من نفسهم حتى بعد كده، حتى if they didn't want.
طبعا دكتور محمد، انا احب انتروديوس فور يو المودريتور بتاع السيشن دي، الاستاذ دكتور منى منتصر، استاذ التقويم في جامعه ورئيس القسم في كليه طب اسنان جامعه المنصوره. دكتوره منى.
دكتور حمدي.
اتفضلي دكتوره منى. اتفضلي دكتوره منى، السيشن بتاعت حضرتك، وان شاء الله حضرتك البرميشن لدكتور محمد يبدا البرزنتيشن بتاعه. اتفضلي.
شكرا. طيب أنا طبعا برحب بدكتور محمد. دكتور محمد حسن, lecturer of orthodontics from South Valley University. His topic is sex-specific differences in mouse offspring skulls due to altered maternal calcium and phosphorus exposure. اتفضل يا دكتور محمد.
Good afternoon. I'm Mohamed Gamaluddin Hassan. You can say a brand new lecturer of orthodontics, South Valley University, Egypt. I'm honored to be here today. I believe coming from South Valley University and meeting all of you, it's really an honor. And as a big fan of storytelling, I would like to start telling you my story today. And actually, my story starts 1,000 years ago when human beings started to understand and appreciate the meaning of a beautiful and harmonious face. If you can see ancient Egyptians, Greeks, Romans, how they were representing the great gods, queens, and kings, they were usually showing it with a harmonious face. But unfortunately, this is not what we see in our daily life. We always see sometimes a convex profile, like most of Egyptians, and even myself, have a convex profile. And sometimes we have this strong characteristic concave profile. And sometimes we have a severe form of craniofacial malformations like cleft lip and palate. And I believe since the establishment of orthodontics, there have been always a debate. Who is taking the upper hand? Is it genetics or environment? This has been always the debate. Sometimes we can have some environmental factors easily identified and other genetics, but sometimes we don't know who is taking the upper hand. And I believe recently it has been shown that environments plays a very important role in our body function. What we are eating, what we are drinking, if you are a smoker, drinking alcohol, or even the stress in your marriage life, it seems it's affecting your physiology. And it was interesting to show you that we published a few months ago an interesting paper about the effect of multi-generational soft diet consumption on the craniofacial morphology. But here today, I'm not talking about the consistency, I'm talking about the contents of the diet. So hopefully today I will answer a question, which is if altering maternal diet contents can affect the offspring's skull morphology, and if could be in a sex-specific pattern. And the way I'm so happy with this project or this topic, it was a really nice collaboration between South Valley University, Alexandria University, where I got my PhD, UCSF, California, San Francisco, where I got my Fulbright fellowship, UMKC. So it was really nice collaboration. So we had an hypothesis here. And I believe our hypothesis is easy, straightforward. We hypothesized that altering calcium and phosphorus levels during pregnancy, it may lead to change the morphology of offspring's skulls in a sex-specific way. And as I mentioned, because I like storytelling, so let's make it easy. What we did to do to test this hypothesis, what did we do? So simply, we brought a bunch of, or a group of female mice. And once pregnancy has been identified, we divided them into two groups. The control group received a normal diet, there is nothing wrong in it, and the experiment group during pregnancy was receiving a special diet with 3 times higher in calcium and 3 times lower in phosphorus. And if you were asking me why 3 times higher in calcium and lower in phosphorus, it's just to keep the calcium-phosphorus ratio the same. We don't alter it. And once they delivered their pups and delivered the offsprings, we moved 2 groups into normal diet. So the pregnant female, after delivering their pups, they received a normal diet. And we waited till the pups received or reached the puberty, which is 6 weeks old. So what we did after this is we collected the pups and we did a micro CT scanning to get a high resolution scanning of the skulls. And then we divided the mandible and the skull differently in a way. And we worked so hard on the mandible, and we published a nice, interesting data on the orthodontics craniofacial research before, because I was so curious to see how diet could affect the temporomandibular joint, and it was really interesting study. But now I'm talking about an unpublished data related to the morphology of the skull. And when we talk about morphology, we talk about the size and the shape, and this is very important to mention the difference, because shape, which is really interesting, it shows the characteristics of the objects. While size, it's a measure of scale. It shows you how the object is smaller or bigger. So to assess the shape variation among groups, we use what's called a principal component analysis and canonical variate analysis. We placed a bunch of landmarks where they can simplify the shape of the skull. And why geometric morphometric analysis? Because simply with geometric morphometric analysis, you can move the effect of translation, scaling, and rotation. So what's remaining at the end, it's only the shape variation. So you can analyze the shape differences. And to analyze the variation in size, we used a bunch of linear measurements that have been used previously in literature. In addition, we used another measure of size, which is a centroid size, getting from the landmarks we used before. And furthermore, it was interesting that we assessed the differences in the tissue mineral density. And I can tell you later after I finish my presentation, because there is a funny story behind the mineral density, but we assessed the density of the alveolar bone in between the roots. So we had the bone between the roots and we assessed the mineral density, so the variation in the grayscale intensity between the groups. So what did we find? And I believe this is the interesting. Regarding the tissue mineral density, we noticed that the offsprings coming from the experimental females had a significant decrease in the tissue mineral density, which was okay, it's nice, interesting. But when we dug deeper and we subdivided the data based on gender, we noticed that the density significantly decreased in females, but significantly increased in males. So here we found a sex-specific variation, which was really interesting. And the interesting, I believe, that the sex-specific changes here have been repeated on the all other measurements. Like, for example, when we assess the morphometric analysis and we wanted to see the changes in the shape, we could see here that is the cluster representing the control is away, or it's clustered differently from the experiment. But when we clustered our data based on intervention and gender, it was really to see that on the right side, we could see the experimental females, but on the left side, we can see the experimental males, which means they have a different shape of the skull. What that means when we are placing the representation of this variation, the females were getting shorter in the anteroposterior dimension, wider laterally, or not wider, but the neurocranium was a bit bigger. But on the other side, the experimental males were getting longer in the anteroposterior, but the neurocranium were a bit depressed. And I believe the interesting that we could see the same sex-specific variation on the size measurement as well. So when we could see the overall change in the size of the cranium and mid-face, yes, there was no significant decrease in the experiment in relation to control. But when we subdivided again the data based on the gender, we could notice that here with the males, we could see that an increase in the size, but with females, there was a significant decrease in the size. And what happened with the overall size differences has been repeated again on the other linear measurements. Yes, there was no non-significant decrease with the experiment, but when we subdivided the data on the gender, based on the gender again, we could see that the male measurement was increasing, but the female measurements were decreasing. So this is nice and great. What's my take-home message here, what we want to say at the end of this presentation. I would say that altered calcium and phosphorus levels during development has a significant differences on the mouse skulls, the offspring's mouse skulls. With the offspring males, it was getting larger or bigger with increased tissue mineral density. But on the other side, the females was showing the opposite effect, like it was getting smaller. And at the same time, we could see a decrease in the mineral density. At the end, I believe, what is the significance of what I'm saying? Like, it looks cool, nice data, it's nice to publish, but I believe in science, and I believe the message of science in with our society. Regarding to the layperson, we want to say here and to emphasize that it seems that lifestyle is affecting our body, but it's not only our body, but our offsprings as well. What we are eating, what we are drinking, we could see a lot of strange posts on Facebook now asking people to take supplements, but no one knows what's the impact of the supplements on the second generation. And this is really a big issue. Regarding professionals, it's really interesting to see more careful design studies about the etiology of malocclusion. And I am a big advocate of the translational research, how to push this basic science data into clinical trials. By the end of my presentation, I would like to thank my mentors. I would like to thank Alexandria University, where I got my PhD. I would like to thank my funding agencies. Everyone helped me to do this work. And finally, I would like to thank you for having the patience to listen to me till the end. I know I am the geek guy who's always talking about mice, but thank you for your patience and your time. Thank you.
Thank you very much, Dr. Mohamed Hassan, for the impressive presentation.
Thank you so much.
Okay, thank you, Dr. Mohamed. And now I invite the judges if they have any questions for you.
I have a question, actually.
Yes, please.
You, in your title, in the abstract, you mentioned sex-specific differences. Why were you biased at the beginning before performing your study and put this into the title? Whereas you were just looking for that, you didn't know whether there will be a difference or no.
This is a big story I would like to share. So when we designed the study, we didn't know at the beginning there are going to be a 6 specific changes. This was a part of a big grant submitted to NIH in the United States.
I congratulate you.
Oh, thank you. Thank you for your nice word. So it was a big study, and this was part of our, how to say, a supplementary data. And we wanted to use the supplementary data to submit another NIH grant, but a smaller grant. It's called RO3 grant. So we were just looking to how diet is going to affect the morphology at the beginning. But we didn't know there are going to be 6 specific changes. And the idea, I will tell you the funny story now. When I was segmenting my mandibles and skulls, I was noticing always there is something wrong related to the thresholding where I was segmenting my data. And the thresholding was always kind of troubleshooting with females. I was always seeing something weird. So we started to look deeper to see if there is a 6 specific changes. And finally, we found a 6 specific changes. So we wanted to say why we shouldn't introduce this mouse model in the literature as something new that has not been mentioned before. And I believe this is what we are trying to do now. Like, we are trying to expanding this, and I will not say a lot of confidential data, but for example, we are trying to see what's the epigenetic markers that could be changed with the change with the diet. What's going to be the, for example, the hormonal changes that could lead to this specific changes. So we are working on this now, adding more biochemical issues. We are trying to make more bioinformatics related to the epigenetic markers like the methylation groups, the histones groups that could be impacted due to the changes in our diet regimens.
I'm impressed, Dr. Mohamed.
Another
question, please. Why did you choose the FVP wild-type mice?
This wasn't my selection, to be straightforward with you. This was my mentor, and sometimes it's based on the available mice in the vivarium. So I believe he had some good mice in the vivarium, and he didn't want to buy new ones because sometimes it's a bit expensive. So he had the FVP. Sometimes we use the C57 Black Jay, but actually for both, it wasn't a big issue in our study.
No, I'm sorry, I will correct this because there are certain species that could be accepted for certain studies and others are not. So please, you have to refer this into your research, because this is one of the very important things. If you use the wrong species for the research, it might not give you the data required. So it has to be explained that it has been documented before that it was used under this conditions, so that it can be accepted in your study. Thank you.
Good question. Thank you.
Doctor.
I have a question, if you don't mind, please, Dr. Mohamed.
Yes, please.
Yeah, you changed the calcium and phosphate level in diet, yeah?
Yes, the calcium and phosphorus, yeah.
Yeah, I would expect to see a difference in bone density, but how does calcium and phosphate will affect the morphology? Can you elaborate more on this point, if you don't mind, please?
Yeah, so we had an issue before, or the bigger study was how can altering osteoclastic activity will lead to change in the craniofacial morphology. And we wanted here to target the osteoclastic activity or bone turnover through different methods. So one big arm of the big study was altering the bone turnover by using knockout mice, where we knocked out specific genes related to bone turnover. And the other one was using injection of siRNA against CTSK gene related to osteoclastic expression. And the third one was targeting the bone turnover using the diet. So the question, I believe, would be nice if we say how we could be sure that the change of the pathway of calcium and phosphorus through the GIT will lead to a specific change in the serum, or the uterine from the mother, like circulation to the fetus circulation. Would be more interesting, but because this was out of my scope, I just wanted first, in the first grant, to establish the mouse model and to establish the morphology. And then I can take this preliminary data to apply for a further grant where I can expand how the pathway could lead to the change in the phenotype.
Thank you, Dr. Mohamed. Thank you.
دكتور محمد انا بس عندي اتفضل يا دكتور منى.
اتفضل دكتور حمدي.
Just two comments. انت طبعا قلت I like يعني is a big fan of transitional research. Okay, how you will go for transition of this data? Okay, maybe something like a survey, retrospective studies, or something like that. Okay, so I would like to know how you will translate this, nice data, because the first publication you talk about the mandible, the second about the skull, all of this in the mice form, how to translate this data to human. This is the first one. The second one, maybe please accept, because I'm coming from the British way in writing, and this is usually conflicting between American and the British. Okay, so I would recommend to make it gender specific instead of sex specific. I mean the title. It's up to you. I know this is a conflict between American and British.
No. I will tell you another funny story related to this topic. So again, as I am a big fan of storytelling, I usually, when I publish a paper or a manuscript, I make it like a story on Twitter. There is a big trend now in science. It's called science communication on Twitter. So when we talked about gender-specific changes, many people attacked me and told me gender is related to human. But when we are talking on animals, we cannot use the term gender. We use the sex-specific changes. But if we are talking on human beings, yes, I will be using the gender. So this is regarding to the use of the term sex-specific versus gender-specific. The second point, which is really amazing and interesting, there was a couple studies published in a journal called Bone, and Bone, it's really nice article. And there was a study has been done by a British group in Gambia, where a pregnant women had a low calcium intake, and they were giving them a calcium supplementation. And interestingly, they showed our same results, but not on the skull. They were checking the bone length, and they were checking some physiological measurements. That's why I'm telling you, I believe there is a sort of connection between our data and their data that has been done on human beings, because they were showing, for example, that the alteration of calcium supplementation for the pregnant Gambian women led to change in the, for example, the insulin-like growth factor 1, which is kind of related and responsible for growth. That's why I have a strong feeling, but I didn't do it yet, that there is a sort of connection here between the supplementation, the dietary supplementation, and the hormonal expression in the blood or serum. But that's what we are trying to do in the next grant to make more biochemical issues, but it has been established. That when I want to say that physiological parameters in human beings related to calcium supplementation has been established to be affecting the long bone and the blood pressure in a sex-specific way.
Thank you.
Thank you.
Thank you.
Okay, any further questions?
I just have a final comment. I would like to thank Dr. Mohamed again, because this research has given us a broad approach to extensively study a lot of other parameters other than what he did. And it's going to open a door to a lot of data related to these environmental changes and effects. So thank you for sharing with us this information, and something that added to me I'm not in the field, but it added a lot, and it gave me a lot of idea in my field as well. Thank you very much.
Thanks, Professor Fawzi.
Sorry, Dr. Mona, may I take like a few seconds, please?
Oh, okay. Yeah.
Dr. Mohamed, I'm sorry, I noticed in your result you've used we several times. So if you change this, it will be great. If you don't mind, please. Yeah. Thank you. Thanks, Prof. Mona.
No, you're welcome.
I'd like to comment about this again, Dr. Mohamed.
Questions.
You know, I know this is American way in writing, so please
Yeah,
don't
because I'm coming from British as well as you.
They write as they speak. Okay, so I totally agree with this point.
They may concentrate on this point during submission of to IADR. They always using American language, actually, not British.
Yes.
No.
Yes.
American writing language for American journals is different from European journals or international journals.
I
That's
agree.
right. Yes. And even submission, I mean,
Yes.
submission of abstracts as well.
No,
I
because
second
usually
Dr. Mohamed.
Denton in British they write with E. Denton when American. So
That's
I
right.
think they are specific this in IADR. But
That's
I know
right.
that they would like to write we, and it's our results show that. I know this, so I don't blame you about that.
But we can always have it our Egyptian style as well.
Yeah.
It's matches Americans.
Dr. Mohamed, I have just like a few questions or two.
Please.
Okay, first, what was your sample size for how many in each group?
This is a very good question. I have 10 in each group, 6 females and 4 males, and this is not going to be a big sample, but I will tell you 2 issues. The first one, it was agreed and accepted by the reviewer panel in NIH because we expecting to expand the results later. So as a preliminary results, this could be accepted. But at the same time, this is what I like always to say: we need to be critical, not generalizing our data. We are saying that it's a new mouse model, but further investigation definitely will be needed. But there were 10 in each group.
Okay, because if you divide them into male and female, it's a very small sample size.
I agree.
Okay, I want to ask you about why did you choose to change 2 elements like calcium and phosphorus? They are 2 dependent elements. How can you define the changes came from calcium or phosphorus? Why you didn't concentrate on one element? Why you combine 2 dependent or 2?
It's a nice, tricky questions and critical at the same time. I believe in the metabolism of calcium and phosphorus, they were always linked to each other. Okay, so definitely changing in calcium and not changing in phosphorus will lead indirectly to change the phosphorus as well. But in our model, we wanted to keep the calcium and phosphorus ratio to be constant. So increasing the calcium and decreasing the phosphorus with the same value will keep the ratio between them constant. Okay, but definitely it will make so if changing one will not changing the other in a different way, it will be the same ratio between the both. but it makes sense that we need to work on each one's differently. But what we noticed in literature is that most people were talking about the deficiency in calcium, but not the excess in calcium. So we wanted to make something different. So what we want to say here is that we wanted to change the calcium and phosphorus, but we keep the ratio between them the same in the blood.
But maybe, but your hypothesis is based on changing the ratio, because you are looking for the changes in the dietary elements. Like, so if you change the ratio, it's valid to change the ratio, because this is the whole point.
I would say definitely we can do next doing a comparison between increasing the calcium by itself, decreasing the calcium by itself, and playing in phosphorus. But if we go to literature a little bit, we will always notice that the deficiency in phosphorus or the plain phosphorus is not that common like the deficiency or problem in calcium. But yes, your point, Dr. Muntasser, it's so valid. I believe you are right.
Okay. Why did you choose 3 folds? Why not 2 or 4 or 1?
It's a very nice question. It's arbitrary. We didn't have any reference for this. We did it by itself. We tried 3-fold increase and decrease, but there was no reference for this.
Okay. Okay, one comment on the abstract. I noticed in your result that you wrote it in a very general way. Like the presentation was much better, because you in the abstract you mentioned like smaller, narrower transversely, taller vertically. So there is no specific numbers or measurements or anything, and even there is no specific areas or points or structures or anything. So I think the part of the results in the abstract needs to be rewritten again. It was much better in the presentation. Yes. This is
forgive me, like as I mentioned, I'm a brand new lecturer, so that's why I'm learning from professors like you, and I believe if I'm definitely I will change it.
It was the presentation was much better.
Thank you.
Okay. How do you explain the difference in response between the males and females? Because the same change, the same you standardized everything. How do you explain the opposite response in males and females to the same change?
So what has been happened with the clinical study on Gambian females, they noticed that before the calcium supplementation, the insulin-like growth factor 1 in females are higher than males, okay? But with the calcium supplementation, it has been reversed. The insulin-like growth factor 1 has been decreased in females, which led into decrease in the sizes of bone of females. So I cannot tell you that's exactly what's happening in long bones, but it could be the same here, like changing the insulin-like growth factor 1, which is one of the responsible for growth, could be leading to the sex specific differences, because it has been found to be decreased in females and increased in males.
Oh, okay. Just another clarification. You mentioned in your title maternal period or time, maternal calcium phosphorus exposure during the maternal part. So why did you extend your study till the time of weaning? So why you didn't stop it at the because it's like I think it's contradictory. Okay.
I will tell you the idea behind it. So we gave the pregnant females the alternate diet, and then we stopped the experimental diet at delivery. So after delivery, during weaning, both groups having a normal diet. So the change wasn't during lactation, the change was during pregnancy. And after delivery, both groups are under the same conditions during lactation. And then we waited till they reached the puberty because we wanted to see what happened when they become adult.
Okay, but now you have like a leeway time where they were exposed to other environmental factors. So this is our, you might have like many confounding factors during this time affected your results.
Based on the experimental design, as much as I know, but I cannot tell you 100%, they are under the same, and they are in the same vivarium, under the same cycles of light and temperature and everything. What has only been changed is the diet. I would say, if the question could be interesting, is altering the diet to the experimental female will lead to sort of a transitionary shock, okay, to the offsprings, okay? Could be interesting. The change of the diet to experimental group after having an experimental diet, they may lead to shock to the mice. But the question, which is really interesting here, is that the 6 specific changes remained even after having this shock, and it was supported by the clinical trials. So it means that our data is kind of matching the clinical data, or the data has been published before on human beings.
Okay, but you cannot apply the results here with the results on the clinical studies. It's different, so you cannot like match them or connect them, or
I agree.
something
but it was in the same trend. It was in the same trend, but yeah, definitely I cannot compare.
Okay. I think Dr. Hamdi asked you about, I think it may be the same question. What the impact you expect from your research on orthodontics, clinical orthodontics?
I have a lot wants to change in orthodontics. As the orthodontist, you will always say, it's never like braces and wires. And this is the issue. Sometimes you can give the patient the same appliance and the same thing, and it doesn't work for one patient and works for the another. Yes, both have a concave and convex profile, but maybe the origin, it's different. Maybe it's this patient due to the genetic, and this one's due to the environment. It's absolutely amazing to see how the etiology of the malocclusion can affect the outcome of the treatment. Sometimes we don't know what's happened to the patient. I applied the same treatment, same mechanics. Why it happened to work with this patient and didn't work? In my opinion, I will not talk too much. I believe malocclusion is just a sign and a symptom. It's not a disease by itself. It's an expression of something happened at the behind. We need to identify what happened at the behind to be able to be treating it properly.
This is why we are going to precision orthodontics.
Exactly. And this is what I believe in the future is going to be how to be able as an orthodontist to use biomedicine. I don't want to be in the future to be looking to me as the one who gets money from braces. I want to be able to use CRISPR technology, genetic engineering, some stuff like this. I will be able to properly diagnose my patient and identify these factors in my treatment as well. So I totally agree with you, Dr. Montasser.
Okay, so thank you, Dr. Mohamed, for the nice presentation and the nice research, and good luck.
Actually, as the discussion is very interesting, we are pleased to hear all of this discussion, and we didn't stop the discussion because of time. Fortunately, we have a break, and we got we are running of time now, and maybe we have to go to the other session. But good luck, Mohamed, in your Dr. Mohamed, in your future life and future researches. We are very pleased you're sharing your research with us, and we hope a good luck for you.
Thanks a lot.
I just have one final comment, please, to add here. I just want you, Dr. Mohamed, to take care that increasing the calcium content in the diet could lead to some serious side effects on the systemic conditions. So please look at this very closely during your alteration of diet, and please look as well to the other elemental alterations that could affect the mineralization of this, and not only the calcium and phosphorus.
I will tell you a final secret, and I will run away after this, and definitely altering calcium and phosphorus will affect also the muscular traction and the muscular activity, which might lead to change in the skull and the mandible as well.
And the heart.
Yes.
دكتور محمد الاول انا بشكرك اولا انك قريت الروبريكس كويس جدا تمام وان انت مشيت ده اللي عمل امبروفمنت على فكره ما بين الابستراكت والبرزنتيشن ولو هتشكر حد اشكر الدكتوره الهام، لان هي فعلا اللي حاطه الروبريكس ده، انت ماشي عليه كلمه كلمه زي ما بيقولوا يعني. بشكر بجد بامانه، انا بشكر الدكتوره الهام لان هي guide you to improve your presentation a lot.
انا اللي بشكر حضراتكم على البري سيشن ديسكشنز اللي عملناها اللي ادت الى الفروتفul ريزلتس دي، وبشكر الكانديديت very much ان هو he took the time to look very deeply into that. Thank you.
هو أنا الحقيقة برضو بشكر الدكتورة إلهام مرة تانية الحقيقة، لأنها برضو أشارت إن إحنا نبعت لكم الروبريكس دي in a suitable time for you. This is يعني not only for our judging but also for the participants to get more information and be ready. Actually, thank you, دكتورة إلهام.
Thank you very much, Professor Mohamed. ويعني we really very much appreciate all your efforts till the last minute. Yeah.
Thank you.
احنا طبعا
We have to leave now and go to the other session, I think.
Thank you.